Thanks so much, Nicole, for that really kind introduction. And I just want to shout out here, both Nicole and Allie Cliff, who's in DSP. We wrote these slides together. I had a framework for the the original lecture, which is actually pretty old. Like some version of this lecture has been around for about 11 years at this point. I co-developed it with one of my coding mentors, a woman named Anna Battenhouse, as a graduate student. So so this has been, if you were at the Broad retreat, and you may have seen this in the retreat booklet, this will be very similar. Unfortunately, I don't think in 50 minutes we can really do a live code demonstration justice, but I will give you a QR code to a public access Terra workspace that Nicole and Allie and I also co-wrote together that walks you through pretty much everything that we talk about in this presentation, with the exception of eQTL analysis, and I'll give you some resources for that as well. And as for questions, just throw questions out, and we can take questions from online as we go through, with the caveat that, this is 44-ish slides. I'm going to try to go at a good pace, but also try not to go too fast. So we should have some time for questions as we go, because there'll be topic changes and things like that. Okay, so a little overview. We're going to talk about, just like RNA-seq in general. We'll talk about alignment, aligners and pseudo-aligners, and some basic analysis and quantifying the RNA-seq data. And then I'll give you like 4 slides on eQTL analysis. Just like enough to give everyone a sense to, if you're thinking about doing an eQTL analysis, what are the big variables that you need to keep in mind? Okay, so just a brief preview. There are so many file formats. It's just absolutely ridiculous, and you'll need to learn interconversion of those file formats. And that's not hard, but if it's not something that you're used to doing a lot in your day-to-day work, there is a learning curve. And at some level, we call these general aspects of needing to move from file format to file format and transforming things, we call that data wrangling. So you can harmonize and clean data into a new format. And then finally, the learning curve for this can be pretty steep, particularly if you don't have coding experience, even writing web pages, even basic scripting experience. That gives you a big leg up in terms of understanding how to handle these analyses. But I think that using things like Jupyter Notebooks, which is how Terra, which is one of the ways that Terra works or can work, I think that those provide really nice ways of walking yourself through an analysis and understanding what's going on, even if it's not something that you're particularly familiar with. But just keep in mind the first 1,000 mistakes that you make, and you'll make them pretty quickly. I certainly got through my first 1,000 mistakes in just a few months. So it'll go fast. But the first 6 months or so, first year or so of doing this, if you're really immersed in it, it can be really tough and it can be really frustrating, and you will feel like you're just Googling Stack Overflow all the time. And I just want to state for the record, everyone that I have ever met, with the exception of a few like truly blessedly brilliant people, pretty much everyone has to go through that learning curve. So don't beat yourself up for it. It's just part of learning a new skill set. I think that Nicole just found some really adorable pictures. Yeah, you can't quite see it at the bottom, but the artwork is by Allison Horst. I guess it's a data friend. We'll call it a data friend. So this is a very text-heavy slide, but here's some caveats. So it's just, and we'll get into more of these as we go through, but good experimental design, adequate depth of sequencing, high-quality sequencing data, you need those for a robust analysis. You can have the best code in the world, and if your RNA was degraded before you started working with it, it's going to be very hard for you to pull any interesting or anything out of that analysis. And that's not your fault. It's just that sometimes data is of variable quality. So just keep that in mind. This workshop is on bulk RNA sequencing data. I've also taught, and I know that the MPG Primer series also involves a lot of single-cell RNA sequencing data analysis. But this is very specifically for when you take a tissue and you remove or some cells, and you remove the RNA from them, and process it and sequence it in various ways. This does not apply to when you're talking about single-cell analysis, which is substantially more complex. I'm giving a threshold here that is a broad threshold, but I generally try to say that 70 to 80%, something like that, of the differentially expressed genes should be the same across multiple methods. I'm giving you two here, DE-seq2 and EDGE-R, but there's a lot of other ones. And then this is really important for genes that have a p-value or a q-value that's near the significance threshold, which is typically 0.05. So it's just, if you really want, if you're like, I'm so interested in this gene, it's differentially expressed, make sure that it shows up in another analysis. Make sure that that you have robustness in your analysis. And then another caveat here is human and model organisms, so mouse and then rat, and to a lesser extent like pig and zebrafish. Any human and model organisms that have near complete genome and transcriptome references, these are going to be the easiest to work with. It's not that you can't work with non-model organisms. I've made a bear transcriptome, I've made a python transcriptome. You can do it, but that's kind of an add-on to everything we're talking about here. Okay, so here's the workflow for bulk RNA sequencing. So here's all our upstream processes. So you've got your experimental design, you've got your material, you isolate RNA from it, you prepare a library, you do some kind of, it's funny, we call it, it's just sequencing now, but at the time we called it next generation sequencing. And you get your reads back, generally in a FASTA format or a FASTQ format. And so, this is just sort of an exploded out aspect of this, where it's like you got some cells or some tissues, you're extracting your RNA. You may do some polyadenylation selection, or using a primer that has a series of Ts, which we call an oligo-DT primer. You may also use a random hexamer primer. Those are going to influence, there's other ways here too, but those are going to influence the kind of data that you get back out. So if you really want to study small RNAs, you're not going to use the same methods that you would for trying to study predominantly mRNAs. And the analysis methods will change a little bit with that too. And then, so we get our polyadenylated mRNAs, and then we engage in the process of reverse transcription, where we, create a DNA-RNA hybrid, and then we do second strand synthesis to make a DNA-DNA hybrid, and that's cDNA that's derived from the RNA that we've isolated. And then, you add your adapters and your multiplex barcodes and all that good stuff, and then you can, quantify it and put it on the sequencer. So continuing along, the core processes here, we do, and to some extent we don't need to do this as much as we once did, but there's raw read and sequence QC. It's sometimes the program is called FastQC. Those are available on-prem. I think some of them are fairly easy to deploy in a Docker image environment. Just making sure that your sequences, like that something didn't go wrong with the sequencer. There's the Illumina Sequence Analysis Viewer, or SAV. That's something that we can also use on anything that we get back from walk-up, or any other sequencing where you have access to the run folder. And if you want to get real in the weeds about that, just let me know and we can talk about it. Then you go through some form of alignment or pseudo-alignment. You look at your alignment metrics and your QC. And then we have downstream processes. So there's a basic form of analysis where you look at, like, what coverage of genes do I have? What coverage of the transcriptome do I have? What do my counts look like? What's my average, it's like also average alignment rate and how that relates to those things. So there's a lot of basic things that we can do. And we won't be able to get into this as much today, but the notebook, the Terra notebook, gets into downstream analysis and interpretation. So the Terra notebook takes the products of a pseudo-alignment. We'll get into the difference between alignment and pseudo-alignment in a few slides. And we go through differential gene expression, annotation, some principal component analysis, and a little bit of pathway analysis as well. And then another important aspect here is, if you don't have a reference, you have to assemble a reference. So if you have recovered samples, this is a big thing for folks in the vertebrate genomics group, anyone who works with like novel sequences. This happens for microbes too, which is really interesting because microbe genomes are really small, so assembling a novel one is not particularly difficult. But you have to assemble that. There's a whole process for that. And there's a bunch of different ways to do it. One of the most common is Trinity or Velvet. And then you set up your references and your QC, and then you make a reference assembly. And in the case of human, mouse, rat, and a lot of common ones, you will be able to access a reference assembly and a reference transcriptome that has the contigs all nicely arranged and like named as chromosomes in most cases. So the lecture is going to focus on this, And then the Terra notebook, which will not be hands-on, but will be accessible to everybody, will focus on this aspect. Okay, so to switch functions and talk a little bit about what we're actually sequencing here, library complexity is something I think is important to address here. It's a function of experiment type, and it relates to the diversity of the fragments that you have in a library. There we go. It matters because libraries that are more complex, libraries that generally have a lower duplication rate, it's a more representative snapshot of what's going on in the cell or collection of cells or chunks of a tissue that you're working on at a given point in time. So if you have a high duplication rate and relatively low library complexity in an RNA-seq library, it doesn't mean you can't get any information out of it, but it does mean that your input RNA was probably quite a small amount in some way, or maybe you're working with cells that are really quiescent and are not expressing that many things off their transcriptome. There's a lot of reasons why you might have a more or less complex library. Library complexity is also a function of experiment type. So things where the search space in terms of the genome or transcriptome is big, like the whole genome or the whole transcriptome, those are going to be where we see higher complexity. So highest complexity, generally, whole genome sequencing, methyl EM-seq or bisulfite sequencing, which is also generally over the whole genome. And then you go down to things that are like exome, whole exome sequencing, RNA sequencing. These are somewhat similar, though not exactly the same in terms of method. And then you've got things like ChIP-seq or amplicons or small RNA sequencing, things like that. And those have quite low library complexity. And the duplication rate and the sequencing data will, this library complexity is a big tuning function on what your alignment rate's going to look like. So just keep that in mind as you work through your own data. Okay, so I've used some of these words. A lot of you are probably pretty familiar with them. But in terms of read sequence terminology, the cDNA is a double-stranded DNA molecule that has been reverse transcribed from the RNA. We've gone through 2 rounds of synthesis. And then the sequencing library has these green and purple adapters added to either end. I'm giving you an Illumina example, but in practice, going from Illumina to things like nanopore is not, or other interesting sequencing technologies that we have available at the Broad at GP, like the Avidity element, it's not particularly difficult. It's a sequence of DNA that the sequencer recognizes, and that's what allows you to read into this black area here. So the sequencer basically you flow on primers that match these purple primers, and it lets you read into this internal region, which is what we call the insert. And actually, okay, I should have just been pointing at this one because I'm actually talking about that. So for the actual sequencing reads, in Illumina, there's generally 4 reads, and they're the indices at either end. This would be the I5 index in yellow here, and then at this end in green, that would be the I7 index. And then you also have read 1, which I generally think of as being associated with the I7 and being on this side of the molecule, and read 2, which I generally associate with being on the I5 side of the molecule, this side of the molecule. Yeah, and the reads at this day can be up to 1,000 bases and well over 1,000 bases if you're doing things like nanopore or PacBio. But the structure is a little bit different. I did a lot of RNA sequencing that was paired end sequencing 2 by 50, and the data was quite good. I mean, they let me publish it, so I'm gonna assume it passed that particular, aspect. But if you can get longer reads, your ability to align to the genome will be better. So to get at your question from another perspective, and this is actually not just RNA, it's more generic to all sequencing data. If you play around with the UCSC genome browser, you can get a unique alignment, with a primer pair using 18 to 22 base primers, depending on the part of the genome you're trying to get at, assuming there's lots of repeats. So if you think about that in the context of alignment, you need something that is you don't really want it to be 20 bases long. That's kind of hard. Some people do that for small RNAs, and it is genuinely more difficult there. The library complexity is different. But if you're in the 25 to 30 base pair long reads, especially if you have a read 1 and a read 2, you're going to be able to align just fine. And a lot of the original early pre-Illumina Celexa technology was 1 by 36, 2 by 36. So you can get a lot done with fairly short reads. But in practice these days, we'd rarely go below 50. Okay, so So just to continue with the terminology here, and please keep the questions coming, they're great. So DNA fragments are the molecules that you sequenced, and the reads are the sequences you got from the sequencer. So the fragments are like the physical thing that you amplified, and the reads are the character sequence and the data that you've obtained, once you've gone from the pictures that Illumina sequencers generate, or the electrical changes that like Oxford Nanopore generates, and those have been converted into a character sequence. You can play with the upstream stuff, but that is really interesting and also fairly complicated. Okay, so yeah, so you're basically getting a readout of all or part of a DNA fragment. That has to do a lot with the read length that you're going for. And then for paired end sequencing, you sequence either end, as we discussed here, and then an R1 and an R2 form a read pair, because in Illumina sequencing, there is this like bridging. We're not going to get into this in detail. There's a lot of really good videos of it, but there's this bridging aspect that occurs where you start with the P5 and then you like bridge over to the P7, and that's how you get a second read. And that wasn't a very good picture I made with my hands, but Illumina made really nice videos that like really show how that happens on the flow cell physically. And read pairs are nice because they let you get an understanding of the average insert size, the size of your, of the DNA fragments that you put in between the adapters, which can be a useful thing to know in terms of thinking about sequencing alignment. Okay, so this is a really important piece, and really, like, this is not necessarily just for RNA sequencing. This is really for a lot of DNA sequencing. Sequencing reads can represent unique or duplicated DNA fragments. So if we have 3 DNA fragments, which ones are duplicates? So the truth here is that we have these sequences, and these 2 are duplicates. But how would the sequencing structure affect this? If you have single-end sequencing, you won't actually know. You'll think that they're all the same thing. If you have paired end sequencing, you can actually more accurately determine the duplication rate. I'm not going to say that single end sequencing is like not useful. There's a lot of good use cases there. Maybe you need a single really long read for your particular analysis. But just keep this piece in mind, that if you really want to get accurate about duplication, if that's important for what you're doing, you generally want paired end sequencing. Okay. All right, so now we're going to switch into alignment, aligners, pseudo-aligners. And just to put you into place, we're working on this section, and there's actually a whole alignment workflow. So like most of those single boxes explode out into like huge flowcharts. And so here, you start by obtaining a reference assembly. We'll talk about that. And there's, and that'll be in a FOSTA format typically. And then you need to build an aligner-specific reference index. And just like it sounds, they are specific to the aligner. So every aligner you want to work with, you're going to have to build a different index. But these days, it's not quite as computationally intensive as it was when I first wrote this 10 years ago. The general workflow is that you would QC and trim your raw reads, these sequences that you've gotten to you from walk-up or from another sequencing facility. You'll align those reads to your reference. There's, align and then there's like, we're giving a couple of commands here. But you get sequence alignment format, and it's called SAM format, and that gets converted into BAM format, which is SAM format, but it's compressed, so it doesn't take up a massive amount of space, just a lot of space. And then you sort the BAM by position. This is, again, one of those things that used to be pretty difficult and is substantially less difficult now. You can handle duplicates. I'm mentioning Picard because it's a Broad-generated tool. There's other ways to do duplicate marking. Some aligners these days will do that for you if you put that into your alignment. And then you'll index the BAM so that you can, like sort through the BAM, and you generate alignment metrics and QC. And there's a couple of nice ways to do this with SAM tools. We're going to walk through all of these in more detail. So in terms of obtaining your reference. So let's just back up. What's a reference? It's a series of named representative sequences. They're versioned. So like I can tell you that I worked a lot with like the pig genomes, or the pig genome sequence version 11. That's sus-scrofa 11. And I work a lot with the human genome sequence 38, GRCH38. These names have changed over the years. The naming sequence recently changed. So if you see stuff with HG19, that is quite a bit older and is about it. That reference sequence is about a decade old and occurs before this naming convention changed. So you can have a genome, you can have a transcriptome. Genomes are generally in FOSTA format. Transcriptomes are generally in a format that's called gene transfer format or gene feature format, and it's a tab delimited format. And you just want to make sure that your transcriptome is the same version as your genome. So if I use an HG19 transcriptome and an HG38 genome, I'm not going to have a good time. These are useful because these are standard to compare and analyze genetic information. There's a lot of good references available from Ensemble, UCSC, GenBank, GenCode. Prokaryotes and microbes, you can get a lot of references from NCBI. And then there are times where you'll have a consortia generate a custom reference. The Bayer genome I worked with was the Ursus arctos horribilis genome reference consortia, which is kind of cool. Okay, so then you got to build your aligner-specific reference index. So these indexing, which we do for the aligned BAM file and the index that we use to align that BAM file, it's basically a way to be able to page through a massive file without losing a massive amount of computational efficiency. Yeah, genomes are gigantic, and it's more memory efficient to do it this way. This is not as big of a consideration if you're doing a lot of microbe stuff, because their genomes are on average like 4 to 10 million base pairs. So throw some of this out the window if you are doing microbial genetics. And generally, what an index takes for transcriptome-aware aligners is a little different if you're doing if you're doing DNA-only aligners. But for transcriptome-aware aligners, you'll have a FOSTA and you'll have a GTF, and there's a aligner-specific index command. And then you'll have some kind of aligner-specific formats. Like for Callisto, it's .idx. I'm trying to remember what it is for BWA, but it's been a little while since I've actually stared at that directory on-prem. But just keep in mind, these are specific to the genome or transcriptome that you're using. You want those to match, and then they're also specific to each aligner. So if you've been using BWA or Bowtie, and now you want to use STAR, you'll need a different reference for each of those aligners. And you just make it once, and you save it. And there used to be a lot in here about how to organize your directories efficiently, and I'm not going to drag you guys all through that, but having like a directory structure that's like references Callisto HG38, references Callisto MM 39 or something like that, or GRCM 39 for the new mouse genome, that will make your life much easier. And I highly recommend just organize your files, organize your life. Okay, so moving into the alignment workflow proper. So for QCing and trimming raw reads, this is important, especially if it's the first time you've done a particular kind of experiment. It's something that you can kind of spot check a little bit, but basically you want to make sure that you don't ignore that. I mean, GP is really good, walkup is really good. We have fantastic people that work here who make the sequencers do really amazing things in incredibly fast and efficient ways. That being said, it's not like they don't occasionally get a bubble on the flow cell. And they usually know that because they do a lot of Illumina, flow cell QC. You want to check your sequences too. I recommend using FastQC, and you basically want to make sure, is your data quality high? Do you have some things like you have really short sequences and you're running into adapters on either end? Some aligners will automatically trim adapters, I learned the other day. And this is useful because garbage in, garbage out. You want to make sure that you're dealing with good quality, reads and data, and not marginal quality reads and data before you invest a ton of time into your analysis. So this is like the first step. And then FastQC, there's some other tools too, but FastQC is on-prem. I think there's several Docker images where you can access FastQC on Terra as well. Okay. There's also trimming of raw reads. And so what I was talking about is, if you have a short DNA fragment and you have a long, R1 or R2, you can read into the adapter on either side. And not all aligners, STAR will automatically trim this for you. Not all aligners will automatically trim this for you. And there's CutAdapt does this, and there's a few other trimming programs, and it seems increasingly because these sequences for Illumina are so well, these adapter sequences for Illumina are so well defined, increasingly it seems like we're just kind of cutting that out of the process. And we're removing a little bit of the human involvement in this process, but check the documentation for your aligner before, before just ignoring this step. And also check your QC before, you decide whether you need to do this or not. All right, so now we're gonna align the reads to the reference. So because we're by and large talking about Illumina data, I'm going to talk about short read aligners. Again, if you're doing stuff with Oxford Nanopore, they have their own aligner, and BWA actually does have a long read mode. I did help someone out with that a little while ago, so I can tell you that. And I know that PacBio has a fantastically detailed GitHub that I think includes like a whole vertically integrated pipeline for all of their stuff. But for Illumina, there's a variety of short read aligners that you can work with. So these determine the placement of your reads or query sequences against a known reference, which is that genome and transcriptome that you indexed. And you just want, you just want a few or the best mapping. So yeah, basically if you have something that aligns to a lot of different places, you're gonna want, but it's like, this one's, kind of okay quality and this one's really good quality, you want that one. And so there's different aligners handle that in different ways. I'm mentioning some of the popular ones. You've got STAR, HiSat2, Bowtie. I think STAR is really great. From the time that it was developed about 11 years ago, it's a really fantastic tool. And the other thing that I always advocate when you're going after aligners, there are humans behind this process. So you want to pick tools, not just aligners, but tools in general that are where you have people that are dedicated to maintaining those tools. That will make your life easier. Just a little bit on the STAR alignment schematic. Here's your query sequence. And STAR allows you to do things like, say, this part of this read aligns to this exon, and this part of this read aligns to this exon, things like that. You can do gapped alignments. Paired end, okay, I've talked a little bit about this verbally, so we'll go quick. But paired end alignment improves mapping because once you anchor one end, the search space for the other end is dramatically reduced. So that's a really nice aspect of paired end sequencing. You can also get, so there's 3 outcomes for paired end sequencing. You can get a singleton or an orphan where one end of the pair maps, because again, the R1s and the R2 are linked from the sequencer itself. And that is reflected in the FASTA data and is also reflected in your alignment data too. You can have, this is the thing you want, you can have what's called a proper pair, where you have, the reads, read 1 and read 2 aligned within some reasonable insert size. And there are usually parameters set for this, where, if you're beyond a certain distance, you have the read 1 and the read 2 mapped, then you'll go from being a proper pair to a discordant pair. And that would indicate that the reads are kind of, it's like you have one facing out and one facing out. They're aligned in a way that you wouldn't expect given the insert, given the way the insert is set up, the DNA insert is set up. And then the insert size is also in the alignment record for proper and discordant pairs. This can be really, without going any further, this can be really, really interesting for particularly changed cancer genomes. Okay, so aligners and pseudo-aligners. On-prem, we've got Bowtie 2, Top Hat 2, HiSat 2, and STAR. There's a few other pseudo-aligners. There's Callisto and Salmon. I'll talk about pseudo-aligners in a minute. And then on Terra, you can go through the Terra, you go through the little hamburger menu, and then you go to the Terra library, and then you go to workflows. And this is really cool. So some people have written these things for Terra. So if you want to get these things up and running on Terra, there are ways to do it. So there's the doc store and the Broad Methods repository. Okay. Broad Methods repository, you can find additional methods. And you can see that Callisto bus tools, which is the single cell Callisto, and then, running Callisto. Those are both things that other Brodies have made, and we can access them through this repository on Terra. So a Callisto is a pseudo-aligner. It's extremely fast. The index build is well under an hour, though if you're doing things with pre-mRNA indices, it's quite a bit longer because the search space is bigger. It can quantify 50 to 70 million reads in an hour. On-prem, there's several versions of Callisto. And also I just want to say, Lior Pachter, who developed Callisto, he very actively maintains it. His lab very actively maintains it, and they have an extensive list of tutorials and guides, which is something that I think is a real plus. Okay, so to switch into sequencing data like sizes, you use the SAM and BAM format basically everywhere. I know there's a few new ones, but use SAM and BAM to store and access data efficiently. So they store sequencing data. This was co-developed by Brody some number of years ago. And we have headers and alignments. As I mentioned, this is a standard format. It's if you're going on like Gene Expression Omnibus, or if you're going on, if you have access to data sets on dbGaP and you want to reanalyze them, you do have the option a lot of the time from going from the primary BAM file. And you can deconvolute that into FASTQ files if you want to realign it entirely. But it's a standard these days. And it gives this header, and it gives data about how the alignments were produced. Usually, these days, it often includes like the whole command of the alignment that was run, which is very, very helpful as a trail of breadcrumbs to help you understand what the other person was doing when they aligned that data. And then it has the actual alignments, and it gives data about those alignments and how well these individual reads aligned. So basically, SAM files are gigantic. They are human readable. BAM files are much smaller. They're not human readable. So like on a SAM file, you can just use Unix tools like more, cat, and less, because those files are not, they're not compressed. And in BAM files, you'll need to pipe through something like SAM tools in order to view those reads, but that's not difficult. Okay, so now we're going to go through like this sort of like, you've got your alignments, and how do you get them into a form that you can do additional downstream analysis? So you need to convert and sort. So we do SAM to BAM, and some aligners will do this for you automatically. And it's part because we don't have a lot of space. Space is a consideration. And so how do we handle this? All right, so I'm going to start going a little faster because we are getting down to it on times. But we're going to basically, you basically generate these files, use SAM tools view, SAM tools sort. They're fairly, they're fairly easy to understand tools. The manual for them is fantastic. And then you also have to decide how are you going to flag duplicates. Flagging duplicates is important because you don't want to double count data. This is why paired end sequencing is really important. So at a minimum, you don't necessarily want to remove duplicates, but you definitely want to flag them. Some people might want to remove them, and there's options for that. But you can also use the tool Picard mark duplicates. And then we've talked about indexing for the indices themselves, for the genome indices themselves, and so too for alignments of data that you've generated. You need to be able to move through the file rapidly, and thus we need to have an index to move through that file rapidly. Okay, finally, some alignment metrics in QC. So you want to know how well the data aligned. Some aligners will output this for you in a log file, which is really nice, but not all of them will. And you can use tools like SAMTools FlagStat to get you an understanding of how many proper pairs did you have, how many discordant pairs did you have, what was your alignment rate. And then there's also RNA-seq QC2, which is a Brody developed thing. And then you can also, this gets a little more complex, gene coverage, bias, strand specificity for anybody playing around with that. So those are useful tools to get at. Okay, so now we're going to get to some more downstream basic analysis. Okay, so alignment just gives you the aligned regions for a given genome or assembly. We need to quantify these, and we generally specify what we want to quantify over for transcriptome alignments using the GTF or the GFF file. And so here's the thing, you can take a GFF or GTF, you can drop yourself into R or whatever your favorite, like systematic manipulation program is. And like, if you have a region that you think is expressed, or a particular intron or exon or whatever, you want to just know how many reads show up over that, or let's say you put GFP as a reporter and you want to know how expressed it is, just add that entry in. You can totally do this yourself. And I think that that's really cool and really powerful about this format. It's easily human manipulable. Okay, so now we got to quantify that data. Some aligners have a recommended quantifier. I really like the text import package because it can import quantified data from many, many, many aligners into DE-seq2. And that handles your downstream processing. I'm not saying this is the only way to get it done. I'm just saying this is a way that I really like to get it done. Okay, and then we got to process and normalize the data. Many tools do this, DE-seq2, EdgeR, cufflinks. There I'm sure are a lot of others, but these are the ones that I'm most familiar with. And what you're getting is a normalized matrix of values. And so this accommodates things like gene length, which is a newer one, and sequencing depth normalization, which is one that's been around for quite some time. And you basically you want to be able to compare, like, if a shorter gene is going to, on average, accumulate more reads and be easier to amplify, depending on the size of your inserts, and a longer gene will have less of that. And so you do want to take that into account because gene length, particularly in eukaryotes, can be extremely variable. Okay, so now I'm going to give you a couple slides on downstream analysis and a couple slides on eQTLs. And if we have any extra time, we will do some questions. This has been really fast, but I do have a workbook at the end that you can work through on your own pace. And I think this has set you up fairly well to do so. So this is the punchline of the last like 30 some odd minutes of talking. You want to identify your differentially expressed genes. So there's lots of tools available, EdgeR, DE-seq2. I'm partial to DE-seq2 because I really like the documentation, but some people really like EdgeR. They use slightly different methods. I would say the results tend to be highly concordant across one another, and that is absolutely a test worth doing. You input this normalized expression matrix, and the output is genes with significant changes in expression across a condition. So you can get like fold change, and you'll get some kind of a p-value with a particular test that's significant. You should be using multiple testing corrections, but you can use non-multiply tested, corrected p-values. Just be careful with that because it can lead you astray. Yeah, and I am aggressive, and so I like the Benjamin E. Hochberg p-value of multiple testing correction. I'm showing you a really beautiful volcano plot on the side. I just think that this person here who wrote this R package enhanced volcano, I just think it's a really nice plot. It works inherently with the objects that come from DE-seq2, and it gives you a sense of, I like that it shows like where your cutoffs are for your log2 fold change, where your cutoffs are for p-value for significance. I just think it's nice, but there are also lots of other great R packages out there as well. All right, so after that whirlwind, I've got like 4 slides on eQTLs because I was requested to have a little bit on eQTLs. This is not my particular area of expertise, but I think it's a really, really cool area because of there's a lot of single cell eQTLs, and there's a lot more genotyping data coming online because it is just less expensive to sequence someone's genome than it was, even a year ago. So the thought of, what is an expression quantitative locus, or eQTL? And there's many other quantitative trait loci, but we're talking about those associated with gene expression. This is RNA-seq. The basic premise is that there's a small nucleotide variant, or SNP, that somehow affects the expression of a gene. Mechanistically, we don't necessarily know how this happens. I've got a lot of ideas, and all of them are speculative. But the thought is that you have expression, and generally you have a genotype, and if someone has, AA, they have fairly low expression. If someone has AC, they have intermediate expression. If someone has CC, they have, or you could be thinking of these as ref alt, and I'm just putting in genotypes here. And generally, gene expression will like increase or decrease across these genotypes. But it's a possible aspect of a mechanism for phenotype-associated loci, or like GWAS-associated loci to cause an effect. And these QTLs, they're not just for gene expression. You could have chromatin accessibility QTLs. I think next week's talk is going to be really interesting in that context. You can have splicing QTLs, single cell QTLs for all of these modes. So, and the reason that we really care about that is, and there's a lot of controversy, I think, or maybe not controversy, but there's a lot of discussion about this right now. Integrating GWAS data and eQTL data, you do start to, this is an older paper from 2016, and I think there is some understanding of like, well, how could you actually have an effect that occurs from a GWAS risk-associated locus? Maybe it could happen through an eQTL, but I think that actually fine mapping these is fairly complicated. But the thought is you have a phenotype, you have transcription, and you have a variant that is causal, and establishing that it's causal is really complicated. So this is like, the quick and dirty version. And this is why people care so much about, the confluence of having an understanding of how a specific nucleotide change could cause a change in expression. The most important thing, and the biggest question I've gotten over a lot of years of kind of watching eQTL analyses grow up, is how many people do I need? And again, you need RNA, this is a dual modality analysis. You need, and ideally you want it all on the same platform because batch effects can really, really, really mess things up for eQTLs, especially if you don't have that many people. So I just want to, this is the web utility. This package is called Power eQTL. I think it's really important if you're thinking about like accessing a biobank, but to do an eQTL analysis, you need some kind of genotyping information that may come in an older style genotyping array, like those that GP was doing through just like a year or two, I think. I think they finally sort of phased that out because whole genome sequencing is just less expensive, and then you're not limited to just the couple million SNPs that are on the chip. You can get a little bit, you have a little bit more power when you do whole genome sequencing based genotyping methods or variant calling methods. And then you need RNA-seq for all of those individuals. So let's say you have a collection of people for whom you have genotyping data and you have RNA sequencing data. How many people do you need? Well, you can't really get it done with 50 people no matter what. Even with a massive effect size, you're never going to hit the significance threshold. But at 80% power, which is where this line is right here, let's say you've got 100 people and you've got genotypes for them and you've got RNA-seq data for them, and it looks really, really good. Like it's 100 people after you do your filtering. You could, with a very high minor allele frequency, so that's really important here, you could pick up some eQTLs in that population. But really, the better powered eQTL analysis, you're talking about like multiple hundreds of people. And that allows you to compensate for dropout, or this genotype chip doesn't look very good, or this person's genome didn't sequence very well. There's going to be a little bit of data dropout pretty much no matter what. And so you have to have, it's like 100 people's like the minimum by and large. And I think, the more people you have, the more sensitivity, the more power you're going to have to actually pick up eQTLs that have smaller and smaller effect sizes with smaller and smaller minor allele frequencies. I would also encourage anyone who's interested in eQTL analysis to explore these in existing data. I'm giving you 3 Broad-specific resources, but there's actually a lot more than this. The GTEx portal is fantastic. They have single-cell stuff now too, and they're getting into some single-cell eQTLs, which is just really, really cool. But look at all of these data that they have, and if you look up a dbSNP ID on the GTEx portal, it will straight up tell you for what tissues it is an eQTL, if any. And it'll give you splice QTLs, which I think is really interesting. We also have the single cell portal. So that would be like, there aren't I think the eQTL piece there is like single cell eQTLs the data is very sparse, and so it's more difficult to establish. But this is also a really interesting data source to look at. And I think the groups of people in like DSP and BITS that are working on this on the back end, they're working really actively to add these sort of like bleeding edge analyses to the data that already exist there, which is really cool. And then we also have a Terra whole genome sequencing variant calling workspace, which is available here. That is a publicly available workspace. You can just search for whole genome analysis pipeline on Terra and it will pop up. So that's what I've got. I know that was really fast. The goal here is to uncover biological insights from sequencing data. You have to go through so many steps to get there. You've got to, actually get the data. You have to actually do all the stuff in the lab, right? We didn't even talk about any of that hardly at all. And then you've got to sequence it, and then you got to analyze it. And so that's a lot. And if you want to do all of those things in a singular person, you totally can. I refer to myself as a inveterate fence sitter between being in the lab, in the wet lab, and in the dry lab. But it's a lot of skills. The learning curves are steep. It does get better. We just did a little bit on eQTLs, and I hope that at least impressed upon everyone that if you're going to do an eQTL analysis, you really need dual modality of data for at least 100 people, and ideally more than that. And then this QR code right here, that is a link to the public access Terra notebook for this lecture. I'm going to go back to this slide, but I want to acknowledge, so you'll be able to scan it in a second. But I just want to give acknowledgements for this. Allie Cliff and Nicole Rockweiler, really the 3 of us worked together, and they did an incredible job on making these slides. These were all black and white text slides when I originally wrote it because because that's how my brain worked. They helped me helped it make it be better and more engaging. We had a testing and feedback team, Zanny Ham, Gwen Torrecampo, Valerie Foley, who helped us with the initial breakout stuff, and that was really, really valuable. And then my mentor and the person who taught me how to code and the original creator of this presentation in 2013 is Anna Battenhouse. She's at University of Texas at Austin. If you ever happen to go there in the spring, she teaches a class on next generation sequencing analysis. It's amazing. Think about taking it. I'm going to leave this QR code up. I know we're about at time, but if you got any questions, I can answer. And thanks to MPG Primers for offering to have me here.